video, we're going to talk about how to call genes. In part one, we will talk about what are the rules for calling genes. And in part two, I will demonstrate how to collect data for calling a gene, how to analyze that data, and how to record your analysis in your notebook, but also in DNA Master Notes section. So what do I mean by calling a gene? Here I have circled the sixth open reading frame that was predicted in a genome, a phage genome. And this sixth gene, GP6, has 6 potential starts. Now the green bar shows the open reading frame predicted by either Glimmer or GeneMark. And this right here where I'm pointing is the start that either Glimmer or GeneMark or both predict to be the correct start. But there are 5 other potential starts. And it's our job to figure out whether or not the computer program has predicted the correct start, or is in fact one of these other starts a better option for this particular gene. Our other job is to look at what the protein encoded by this gene possibly does. What is its possible function? And finally, the other thing we're going to do is look into places in the genome where an open reading frame has not been called. And you can see that as we go along the genome, there is a gene, maybe in different frames, but covering most of this particular section of the genome, with only really very small spaces in between, or no space at all. Yet this first section of the genome, at about maybe 200 or 300 nucleotides, there's no gene called. And so we're going to look at spaces and say, what's going on here? Is there some sort of regulatory sequence here, or is there a gene possibly in this open reading frame that the computer programs missed? So the goal for the course is for you to annotate the genome and determine all the most likely starts and functions for all the genes in each of the phage genomes. The goal for the 2 videos, part 1 and part 2, are for you to learn how to collect and analyze data that will help make decisions about open reading frame starts, learn how to record the data analysis, and essentially your entire thinking process or logic for the particular call that you made for that gene in an electronic notebook. And finally, by modeling how to present a gene call to you, I'd like for you to be able to present your gene calls supported by evidence to your peers and to your instructors. So part of your reading assignment is to know very well the guiding principles of phage genome annotation. And so I'm going to go through each of the rules and see if I can explain some of these rules to you. In any particular segment of DNA, there is typically only one frame. Remember there are 6 frames? Only one frame and one strand is used for protein coding gene. So in a double-stranded stretch of DNA, there should be only one gene. So genes do not often overlap by more than a few base pairs, and there are some exceptions where there are genes that overlap by quite a few base pairs, 30 or more. And this doesn't happen very often. So if we see a really large overlap, we want to look at that really, really carefully to decide whether or not we believe these genes have such a large overlap. Otherwise, we see very, very small overlaps. And in fact, the overlap that we like the best is the Fantastic Four. A 4 base pair overlap is a great sign, perhaps from the annotation gods, that you have found the correct start for that gene. And there's a biological reason why 4 base pairs is kind of magic, and we'll talk about what that is in part 2 of this video. So the gene density in phage genomes is very high, so genes tend to be tightly packed. So if there is a gap between the gene, then it's usually quite small. And we often look for the start that gives us the longest possible open reading frame without overlapping too much, but providing the shortest gap possible. And so if we don't have any particular data that really points to one start or another, we will often go with the start that provides the longest open reading frame. Protein coding genes should have coding potential predicted by GLIMR, GeneMark, or GeneMark SMEG, or if you're working with a Gordonia phage, GeneMark Gordonia, or possibly GeneMark tuberculosis. But start sites are chosen such that not only does your gene have coding potential, but your start site should include all the coding potential for the gene. Again, this isn't true 100% of the time, but if you can choose a start that includes all the coding potential, that's the start that you want to choose. So if there are two genes transcribed in opposite directions, for example, GP19 of Sally Special is a leftward transcribed gene because it's on the bottom of the map. These genes 20, 21, and 22 are rightward transcribed genes. So this is an area of divergent transcription. And where you see areas of divergent transcription, there has to be a gap that's at least 50 base pairs or more. In this case, it looks like the gap is about maybe 150 base pairs long. And that's important because there has to be room for a promoter going in the rightward direction and a promoter going in the leftward direction, and these promoters should not overlap with each other. So in these kind of spaces, we expect there to be regulatory sequences and not a gene encoded in the space. Protein coding genes, in general, are at least 120 base pairs long. Longer is better. And that's a rule for most organisms. However, phage do break this rule. There are examples of known amino acids, known proteins that are produced by phage that are 26 amino acids in length, so even shorter than 40 amino acids in length. However, when we are calling genes that are this short, we really want to be suspicious of any gene that's 200 base pairs or less and have a close examination. One thing that we'll look for is the presence of coding potential in that open reading frame in one of the coding potential predicting programs. Okay, so switches in gene orientation from forward to reverse and vice versa are pretty rare. It does occur, but again, this is another place to pay very close attention. And finally, two easy rules. Every protein encoding gene has to have a stop codon. And there's 3 flavors of stop codons. You should recognize some of these. And every single protein coding gene has to start with initiation codon, either ATG, GTG, or TTG. Now, last semester, we talked a lot about ATG, but not so much about these other 2 flavors of starts. They do occur, although not as frequently as the ATG. And the TTG start is particularly rare, but you are likely to find it at least once in your genome. The other thing that you want to consider for start sites is where the gene is located, and also ribosome binding sites, or you guys learn these as Schindel-Garneau sites. And I will have you guys look at particular scores for ribosome binding sites. How strong is the Schindel-Garneau sequence? And it's something that we'll look up, but it's really something that's more like icing on the cake. It's not a piece of data that we weigh very heavily in making our decisions. Perhaps if we had 2 start choices and the data didn't point strongly to one or the other, and it could be either one, perhaps if one of them had a stronger ribosome binding site, then we might go with that start. But if you have, for example, a start that has creates a 4 base pair overlap with the upstream gene, but has a horrible ribosome binding site, then we just live with the horrible ribosome binding site. The 4 base pair overlap would trump. And so that brings us again to the relationship of the closest upstream gene. Usually, again, there isn't a large gap and there isn't a large overlap. Overlaps usually are about 4 to 7 base pairs in length. Again, 4 base pairs are the most bestest. Okay, finally, the position of the start is usually conserved among homologs of genes. So a gene that's found in one phage, the homolog, meaning a very, very similar sequence is found in other phage, they usually have conserved starts. So you can check to see whether the start for the gene of interest in your phage genome is the same as that in other phage by looking at BLAST data. So this is showing an alignment between what is the query, which might be the gene from your phage genome, and the subject that it matches. And I can see from this alignment that the first amino acid of the subject matches up with the first amino acid of the query. So at least for this particular match, it looks like the start is conserved. But a better way to look at that is to look at all of the homologs together and what start options each homolog has. So each one of these colored lines is a start, and these are all genes from different phage genomes that belong to the same family. They're all homologs of each other. And the blue line that's extra thick shows the start that was chosen for that particular homolog. And you can see that the second start is most frequently chosen. There's this one where somebody chose a start further downstream. And this can also indicate that not only is the start conserved in all the homologs, it's present in all the homologs, and it's the same start that everyone else chose. So no one piece of evidence alone should be used to make decisions about likely start positions. So you can't just look at one piece of evidence. For some calls, all the pieces of evidence are going to point in the same direction. All the evidence is going to cooperate, and it'll be an easy decision to make. But for many of the calls, evidence may point in more than one direction. And so more than one start may look probable, in which case we just have to do the best that we can, and we might have to weigh certain evidence over the other, and we'll have to look at these on a gene-to-gene basis. But the big message here is that you do need to look at multiple pieces of evidence in order to support your call, and also this will be true for making function calls, which we'll learn about later. So if we make a call for a start that has a 30 base pair overlap, which I told you earlier, we don't like 30 base pair overlaps. If the start array data, the coding potential data, and a number of other pieces of data indicate that that's the correct start, then this would outweigh something that seems like that data that wouldn't support the call. So again, we have to look at the big picture and all the pieces of data together in order to make these decisions. So in our next video, I will show you how to collect data to make a gene call, how to analyze the data, and how to record all the evidence and the data analysis and your logic in your notebook, and how you can use that to present your gene calls to your peers.